Hello and welcome back. We'll be starting with globular proteins today. So globular proteins are tightly folded into round shapes, and they have an axial ratio of length to width less than 10, and they are dynamic and they're mobile. So one of the main globular proteins is the heme proteins, and those are a complex of protoporphyrin 9 and the iron cation, and heme is the most prevalent metalloporphyrin in humans. It's found all over the place. It's in the myoglobin, hemoglobin, cytochromes, enzymes such as catalase and tryptophan pyrrolase. The heme proteins are proteins with heme as the prosthetic group. So prosthetic group, if you remember, are permanent. They don't just attach. Fe2 plus interacts with the proximal and distal histidine, and it directly interacts with the proximal histidine and at histidine 93 at position F8, and it interacts with the distal histidine, which is histidine 64, at position E7. And the distal helps stabilize the oxygen binding. It plays a role in the cytochrome redox reaction by transferring the reducing equivalents, and it involves like a single electron transfer between the ferrous and ferric states of iron to break down hydrogen peroxide. And the catalase enzyme is the enzyme that catalyzes the breakdown of hydrogen peroxide. The heme group and the myoglobin serve to reversibly bind to oxygen, and tryptophan pyrrolase detoxes the tryptophan and synthesizes nucleotides. It is a major site of heme biosis is in the liver, where the rate is variable, so it responds to whatever the body needs. Sometimes if iron concentration is high, it will secrete heme to bind it. If it's low, it will not produce any heme. And in the bone marrow, it's a almost constant production. Next is myoglobin. Myoglobin is a heme protein present in usually the heart and the skeletal muscle. And it's a reservoir and a transporter of oxygen, especially in the muscle cells. And it is one single polypeptide chain made of 8 alpha helix regions. It is a polar surface, and internally it is non-polar, which is pretty characteristic of all globular proteins. And besides the 2 histidine residues on the inside that bind oxygen, the interior of the myoglobin is made out of only non-polar residues. It's fairly small, and the alpha helical regions are terminated either by the presence of proline or the beta bends and loops, and it is stabilized by hydrogen and ionic bonds. The histidine, like I said before, always involves an oxygen binding. Like hemoglobin, the heme is in the center, surrounded by nonpolar molecules, and is attached by proximal and distal histidines. Proximal, once again, binds directly to the iron, and the distal, like right here, binds directly to the iron, and the distal interacts indirectly. Alright, back to hemoglobin. So hemoglobin is only on the red blood cells, and it transports both oxygen and carbon dioxide. And there are two forms. There is the top form, which will bind the oxygen, and the relaxed form that will release the oxygen. There is a all or none effect, so it either bind or it will not. The taut form releases the oxygen, and the relaxed form binds oxygen. Hemoglobin A is the most abundant in adults. It consists of 2 alpha chains and 2 beta chains held together as a dimeric unit. And HB Gower 1 and 2 are formed in the first few weeks after conception, and then the synthesis of the heme begins in the embryonic yolk sac, and then it starts in the liver, and then finally the bone marrow starts to produce it. 10 to 12 weeks after pregnancy, up to the first 6 months, hemoglobin F starts to develop, and those are being developed by the erythroid precursor cells in the bone marrow. And about 60% of the total hemoglobin in the erythrocytes are starting to form in the first months and the last months of fetal life. About 60% of the total hemoglobin in the erythrocytes during the last months of fetal life. Strange. Let's edit that part out. HB Portland is also an embryonic hemoglobin. Okay, so hemoglobin Gower 1 and 2, along with hemoglobin Portland, are embryonic hemoglobins, and they develop in the first few weeks after conception. And the synthesis of heme begins in the embryonic yolk sac, and then the liver, and then finally the bone marrow will start to produce it. 10 to 12 weeks of pregnancy through the first 6 months of prenatal life, hemoglobin F will start to form, and it was going to be produced by the erythroid precursor cells, and it is the major hemoglobin present in the fetus and the newborn. And then 8 months of pregnancy and onward, finally HbA develops, and the synthesis takes place in the bone marrow, and gradually over time replaces HbF in the erythrocytes. So after birth, 12 weeks after birth and onwards, HbA2 starts to develop, and it is a fairly minor component of normal adult hemoglobin, but it's still there. And then what is useful about HbA is in a clinical scenario, HbA can be glycosylated into HbA1c. HbA1c monitors your insulin levels over like an average of 3 months. So it is dependent on the average plasma concentration of glucose, and it is used as a marker for blood glucose status in patients with diabetes. And this is much more useful than a simple blood sugar reading. This is really used to analyze a patient's lifestyle, because if a patient eats well and cleans up their diet and starts working out 2 weeks before they see the physician and they get their blood glucose checks, it's going to seem normal. But if you check their A1C, you will see how much they have actually put in the work to exercise and to clean up their diet, or how much they've been cheating. So oxygen binding, the heme-heme interaction of the molecules facilitates cooperative binding, which means that once one oxygen comes in and binds to hemoglobin, it makes it easier and encourages more oxygens to bind, so hemoglobin will be filled up. Hemoglobin has 4 oxygen sites, so 4 oxygens can bind, while myoglobin only has 1. The Bohr effect is an inverse relation, so if a saturated hemoglobin will go to a place that has very low oxygen and very high amounts of carbon dioxide, they will able to be they are able to switch out an oxygen for a carbon dioxide. So in a normal condition, a right shift is bad on the oxygen saturation chart, because right shift is a reduced affinity. It means that it takes more effort to bind oxygen to the hemoglobin or the myoglobin. So the oxygen dissociation curve is going to be hyperbolic for myoglobin and sigmoidal for hemoglobin. Not that important, but something useful to know. The oxygen dissociation curve for hemoglobin is steepest at the oxygen concentrations that occur in the tissues, and this allows the oxygen delivery to respond to small changes in oxygen pressure. So binding. Oxygen and carbon dioxide both bind to iron, but in hemoglobin, it has a much higher affinity for carbon monoxide. So the tense state has these crevices, and so there's various binding sites on hemoglobin, and oxygen and carbon monoxide can bind to iron. 2,3-BPG will bind in the crevices, like here and here in the tense form, and carbon dioxide binds to the amino terminal, so the globulins, and the histidines will bind to the the protons will bind to the histidine that bind to the beta subunit. So 2,3-BPG is increased in chronic hypoxemia and anemia. It binds to hemoglobin in the crevice of the tense form and decreases oxygen affinity. So the curve goes towards the right. Fetal hemoglobin binds weakly to 2,3-BPG and has a higher oxygen affinity. So what's useful about that? When the mom's blood circulates and carries oxygen, the fetus's higher affinity hemoglobin will be able to rip away the mom's oxygen and take it to nourish the fetus. So that's another problem in transfused blood. So transfused blood have a decreased amount of 2,3-BPG, so it has an abnormally high oxygen affinity, and it becomes an oxygen trap. So once transfused blood has been transfused into the patient, it's completely bound, so it can't pick up any more oxygen, and without oxygen, no new oxygen can be transported because of the abnormally high oxygen affinity. It's not going to be released, and the patient actually becomes hypoxic. And to kind of like prevent this, transfused RBCs are treated with inosine, which is hypoxanthin ribose, and that is added in order for it to be released, phosphorylated, and enter the HMP pathway and easily converted to 2,3-BPG. So we will add more 2,3-BPG as a sort of like a competitive inhibitor, right? So you just add more, so the chances of it binding be higher. Okay, so carbon dioxide and carbon monoxide. So carbon dioxide can bind to the amino terminal, forming carbenohemoglobin. Now, hemoglobin has a much higher affinity for carbon dioxide, and it forms kind of like the bluish red venous blood. And carbon monoxide binds tightly and to hemoglobin and forms carboxyhemoglobin, and that stabilizes the R form, preventing any oxygen to bind. And the problem is, since it prevents the oxygen to bind, there is no oxygen, transport, and it does that by inhibiting the complex 4 of the electron transport chain. So, in doing that, prolonged exposure causes toxicity to the CNS and the heart. So, the sources could be like house fires, smoking, furnaces, heaters, locked cars, or methylene chloride. And you treat that with 100% oxygen. So, like competitive inhibition, you just want to add more oxygen than carbon dioxide. So slowly, it will release the carbon monoxide and bind to carbon dioxide. So cyanide. Cyanide is found in the pits of various fruits, such as apples and apricots. It's also kind of converted as a hydrocyanic acid from the combustion of burning acrylic and certain plastics. And you can also get it from smoke inhalation. Cyanide inhibits mainly the cytochrome C oxidase, which is complex 4 of the electron transport chain, and decreases the ATP production. So the clinical features is that the skin turns very red, and it has the clinical features of metabolic acidosis, so convulsions and weakness and fatigue, and then leads to coma and then death. Fortunately, there is treatment now. We are quite lucky to live in a time where we can treat something that was as deadly as cyanide poisoning. And we first give them amyl nitrite and sodium nitrate, so it will form methylene hemoglobin, because cyanide has a higher affinity to form cyanmethemoglobin, and then sodium thiocyanate is given, so react to form a sodium thiocyanate, which is excretable. So once the kidneys have excreted that, methylene blue is given to revert methylene hemoglobin back to normal hemoglobin. So methemoglobin is a condition where the ferric state of iron is bound instead of the ferrous state. So with the ferric state, it can't release oxygen, and the NADPH methemoglobin reductase, that is used to treat it, and it reduces the methemoglobin to normal hemoglobin. So this causes problems because it doesn't allow any oxygen transfer once again, and it has a chocolate brown or a muddy kind of colored blood. So to treat this, you use this is not to treat, but some causes of methemoglobinuria are the nitrate drugs, reactive oxygen species, and mutation in the alpha-beta globin chains. So chocolate cyanosis is the vernacular name of methemoglobinemia, and it is caused by either certain drugs such as nitrates and reactive oxygen intermediates, or it's a congenital cause where there's a mutation in the genes for the alpha-beta globin chain, or the reduced synthesis of NADH cytochrome B reductase, or there is a G6PD deficiency, and that causes a deficiency of NADPH. So the signs would be a chocolate-colored blood, here you go, compared to normal blood, and skin. And the symptoms are shortness of breath, hypoxia, cyanosis, altered mental status, headache, fatigue, and dizziness. And the treatment, fortunately, there is one, is to give the patient methylene blue to reduce the methemoglobin back to normal hemoglobin. So sickle cell has a variety of forms, but HBSS is the most serious form where both beta chains have been mutated. And it is an autosomal recessive disorder, and it is caused by a point mutation where a valine replaces a glutamate at position 6. And the deformed shape of the cell causes the calcium influx, and the cross links of the membrane protein is affected. So the potassium, water, and chloride ions efflux, and that causes dehydration, which makes the sickling worse. So what's the problem with sickle cell anemia? The freed hemoglobin is freed from the hemolyzed red blood cells, and it binds to nitric oxide. And without the nitric oxide, the vessels constrict and encourage platelet aggregation. So the sickles are already kind of problematic because they can't be easily cleared away, but because of their shape, they stick together even better. So that can lead to clots and vascular damage, and it has a problem with obstruction that could cause like tissue hypoxia, and it could cause ischemia and necrosis. So this shows up around 5 to 6 months after birth, and you can do a sickling test with sodium metabisulfite. Earliest symptoms in children is there's jaundice and fatigue and dactylitis, which is a painful swelling of the hands and feet. Some complications could be acute chest syndrome, acute pain crisis, chronic pain, delayed growth and puberty. Factors such as increased exercise or low pH or higher 2,3 BPG levels can precipitate an attack. Sickling, people with sickle cell traits don't necessarily go around like short of breath all the time, right? But once there is increased need for oxygen, then there would be more binding, and the more binding causes the more sickling, and then that causes the problem. So Luckily, nowadays, we have some form of a treatment. We can't cure it, but we can treat it. We treat with hydration. So with dehydration, the sickling is worse. So we try to prevent that with the hydration. And for some people, we give them a daily dose of hydroxyurea to increase the amount of fetal hemoglobin, which reduces the amount of white blood cell and the expression of surface adhesions on the neutrophils, red blood cells, and vascular endothelium. So that that will reduce the chances of vasoconstriction and the neutrophil activity, so there wouldn't be an autoimmune response. So occasionally, during a particularly bad crisis, a person might be, they might necessitate occasional packed red blood cell transfusion. Only that, no plasma, and an iron chelator to reduce the iron load. And these patients generally are prescribed some like antibiotics as a prophylaxis, just to reduce the chances of having an infection by pneumococcus bacteria. And a bone marrow transplant is the only cure. Going back to hemoglobin for a little bit, there is one benefit to having the heterozygous state of sickle cell trait. So with the heterozygous state, half of the cells are going to be normal, and in theory that this protects people who are living in places that are endemic to malaria, because the malarial I believe protozoan can't infiltrate the cell when it is sickled like that. So HBC, hemoglobin C, it is when glucose is replaced with lysine. So it is a chronic anemia, but is not as bad as the sickle cell trait. And this is a trait where some chains have the sickling cell mutation, where other chains have a HBC mutation. This can be asymptomatic until a crisis hits, and a crisis usually follows after childbirth or after surgery. And patients usually have a history of crises since childhood, and they are quite painful. But if they don't suffer from an infarctic crisis, no specific therapy is required. There is another disease called hemoglobin SC disease, and hemoglobin SC disease, some of the beta globulin chains have the sickle cell mutations, while the other have the mutation found in HBC mutation, or HBC trait, and the hemoglobin levels tend to be higher in HBSC disease than sickle cell disease, and may even be in the normal range, but they generally remain fairly healthy and undiagnosed unless they have a crisis. And these patients can live a decently long life. Thalassemia is a hereditary hemolytic disease, and it is characterized by an imbalance in the synthesis of globulin chains. So they are the most common single gene disorder. There's a variety of mutations. It could be a gene deletion, a substitution, or a deletion of only one nucleotide in the DNA. Alpha-thalassemia is when the synthesis of the alpha chain is absent or reduced, and the same goes for beta-thalassemia. The synthesis of the beta chains will be absent or reduced. So if you can see here, these are the healthy cells, and these are cells suffering from thalassemia. And thalassemia is a microcytotic disease, and you can see the cells are like either abnormally shaped or they're smaller than normal red blood cells. So alpha-thalassemia, if one of the 4 alpha globulin genes are affected, it's a silent carrier. If there's 2, it's considered a trait. If 3 are affected, you are considered to have hemoglobin H disease, and if you have all 4 genes affected, it's going to be a non-viable fetus. It will be spontaneously aborted because the alpha chains are necessary to make fetal hemoglobin. So if the other chains are unaffected, they will accumulate. And the tetramers are soluble, but they don't have the heme-heme interaction, so there's no cooperative binding. So in alpha-thalassemia, the synthesis of unaffected gamma and then beta globulin chains continue, and it results in an accumulation of gamma tetramers in the newborn, or beta tetramers. And these tetramers are soluble, but the subunits don't have any heme-heme interactions, so no cooperativity. Which means that it makes it useless to us because there is not going to be any oxygen delivery to the tissues. The oxygen affinity is too high. Beta-thalassemia, there's going to be an accumulation of hemoglobin F and hemoglobin Barts. So this will be manifested in hypoxia, microcytic anemia, and risk of blood clots. And there are 3 different types, depending on the severity of symptoms. There's going to be thalassemia major, which is called Cooley's anemia, and it's most severe. There you get signs of jaundice, failure to thrive, enlarged heart, spleen, liver. And then there is Beta-Thalassemia intermedia, of course, is a moderate effect, and Beta-Thalassemia minor, which is a carrier, and there is no effect. Why is that? It's because in Beta-Thalassemia minor, some beta chains are made, and though they are not enough to require specific treatment, patients generally are healthy enough that they can live normally and healthily until there is a crisis. So hemoglobin E is an autosomal recessive disorder, and it is a single point mutation on a beta chain at position 26, and it exchanges a glutamate for a lysine, and it creates an abnormal hemoglobin, and we call it hemoglobin E. So hemoglobin E is a benign symptoms. There's only benign trait. There's only mild symptoms like beta-thalassemia trait, and in Southeast Asia, Vietnam, Laos, Thailand, HBE beta-thalassemia is the most severe form of beta-thalassemia. So it has a wide variety of clinical disorders, and it has a hemoglobin E, if it has interactions with different forms of alpha-thalassemia, and has a co-inheritance with beta-thalassemia, a condition called hemoglobin E beta-thalassemia is found in a lot of populations in Asia, and it's the most common severe form of beta-thalassemia, and it has the highest frequency among people of the Southeast Asian heritage, which are like, Vietnamese, Laos, and Thailand. So hemoglobin D is more common in people of Indian descent and Pakistani ethnic groups, and it is known to be benign, and these patients will not experience any symptoms, and his or her health is not affected. That's about it for today. Thank you for your attention, and good luck studying.